have what a pleasure ourselves working. So let's see if the mic is working. Is the mic working? No.
Oh, I think
Hi, everybody. Nope, still not good. It's getting better. Apparently, we introduce ourselves. So I'm Sue Carter, otherwise known as Mrs. Porges. Oh, after all these years, that's the first time I've ever said that, isn't it? It's true. Steve and I, he's sometimes called Mr. Carter. He doesn't deal well with it, really. He does not. There are quite a few Stephen Carters in the world. Too complicated. It's a real pleasure and amazing turnout. Honestly, I thought we'd be lucky if we just brought the family, our family members. So you guys are brave, and I hope you get on your plane in time. Okay, and we have 3 talks, and we have, each about 20 minutes. So we're going to talk fast. These young ladies and I are the last hurrah from this wonderful conference. I've been here the whole time. I got to hear Steve talk. I hope, how many of you heard his talk? Great, perfect, that's good, because there's continuity here. It'll take me a few minutes to get to that part, and if we're lucky, we can make it all seem like we know what we're doing. So I'm a biologist, a neuroendocrinologist, a behavioral neuroendocrinologist. I've spent my whole career since 1980 studying oxytocin. Okay, why? It was extremely unpopular in the beginning because when I started, everyone believed that oxytocin was a female hormone with no functions other than birth and lactation. Prior to 1980, prior to my first studies, which were in prairie voles, I won't talk about that, but they were little rodents that formed lifelong pair bonds. I was just doing research, looking for answers to, how should we say, not real questions, just things I was interested in. It's called curiosity-driven. I'm not sure if it even exists anymore. So what I want to do is take you on a very quick trip through 50 years of research, and in doing that, try to help you understand at an even deeper level than those who've gotten to hear Steve and other people talking about this at this meeting. So my purpose is to explore the role of oxytocin in human sociality, which again was not a question when I started, and in the healing power of safety and nurture. Oxytocin's a comparatively, evolutionarily modern hormone. It allows us to be human. It is essential to a process. I'm going to give you a quick teaching lesson today on a construct called hormesis and healing after both emotional and physical trauma. Oxytocin's unique to mammals, but there are oxytocin-like molecules in other species. And it allows the unique adaptive strategies that we use. We're very, very social. We're dependent on sociality. Human existence required. We wouldn't be here today if we hadn't solved the problem, which I call the evolution of a social solution for dealing with stress and trauma in a complex world. We had to have social communication to make this work and selective social engagement. All of you work with people in some way, and you know that you've got to have relationships with clients, of course, with families. Also, social bonds and lasting attachments weren't being studied at the biological level when I started this work 46 years ago. But it really was obvious thanks to the fact that I'd been hanging out with Steve already for a decade, and he was studying the autonomic nervous system. We were really studying the same thing. And these work together, and we won't get through the whole story, but you'll see they integrate sociality with health and well-being. Oxytocin and the autonomic nervous system, and especially the ventral vagal system, are one system. The big problem came when we tried to be social. And so I like to just draw these two circles and say these things, our other is necessary for homeostasis, allostasis, and even what I've been calling sociostasis, which we're going to change that to sociodynamic something. We're still working on the language, but it matters across the whole life cycle. The mammalian nervous system is adapted to work with others, and these attachments and communication are absolutely necessary, whether you call it homeostasis or homodynamic relationships, doesn't matter. Social relationships obviously come in many forms, and the associated sense of safety is the underlying integrative quality. I want to give a couple of quick basic principles. I like this quote, the African proverb, if you want to go quickly, go alone. If you want to go far, go together. That's basically it, okay? Social solutions are slower. They cost more, they're more costly, but they're more enduring, and they're facilitated by oxytocin. Of course, different mechanisms are necessary to deal with quick problems and lasting problems. And that's created a lot of confusion in this area, because people have said, oh, they've done experiments in the moment and tried to generalize to the long-lasting solutions. All of you are probably working with the slow problems. You're working with things that accumulate over a lifetime. Most of you are not fixing a broken bone or a quick problem. So, of course, these things didn't just start yesterday. The whole history of life on Earth involves stress, trauma, danger. Survival's the first law of nature. A molecule called oxygen is necessary for life on Earth. It's at the center of the benefits of the healing power of social connection, but it's also a dangerous molecule. It's volatile. It is capable of causing inflammation. So if I don't get through the whole story, let me just say that if you look at what we know about illness, both mental and physical, at the moment, most everything drills down to inflammatory processes. If you look on the internet and you say, I want to live to be 120, that's supposed to be our capacity, the answer to how to live a long and happy life comes down in a lot of different dimensions to inflammatory processes. Oxytocin-like molecules, and in the case of humans, oxytocin itself helped mammals master fire, master inflammation. And it's due to this day. When things don't work properly, you know what happens. We become vulnerable to mental and physical dysfunctions. So the oxytocin vasopressin pathways become dysregulated. They just don't work very well. So what's oxytocin? It's a neuropeptide. Of course, you hear that all the time. Just means a string of amino acids. It's central to safety, love, longevity, and managing stress. The biological secret is probably another simple molecule called sulfur. And oxytocin has what are called disulfide bonds. Most of you say, oh, who cares? But you do care, because when those are working, then oxytocin can open and close. It's like a hinge on the molecule. Makes it a very powerful, almost like a blanket to go over fire, okay? Oxytocin at first, as I've mentioned, was thought to be only in females and only involved in reproduction. It took 40 years, frankly, and still most people don't recognize that oxytocin is part of everything. It's made in the brain. It affects not only, it's a posterior pituitary hormone, but it's actually a hormone that affects directly the brain, and it adapts, protects, and heals. The autonomic peptide pathways evolve together. The one we're going to talk about is pretty specific to mammals. It supports maternal behavior, overcoming stress and trauma. The first big stressor for every human is being born, and for every mother who gives birth, of course, it's birth itself. Now, I like to point out right now, stress is not always bad. In fact, it's necessary. And that's created another kind of confusion. It's manageable stress that we're looking for, because that can actually have benefits. Stress followed by oxytocin release. The model system is birth. And I'll use this term, hormesis. I did not invent this. I'll explain in a minute. Some of you know it, some of you may not. Birth is under biological conditions, which are increasingly rare. It involves oxytocin, and it's followed by lactation. Hormones then, including oxytocin, act on the brain, mother, child, autonomic nervous system. They facilitate birth. They're necessary for lactation. They buffer both mothers and fathers from stress. But oxytocin does not act alone. A third point, because if you pull up something on the internet, this part of it is often left out, unless, frankly, they cite my work, because I've been drilling on this for 20-odd years at least, trying to explain that you can't just hand wave oxytocin does this or does that. It doesn't do anything by itself. It's always working in partnership with many molecules, but especially vasopressin. They evolve together. They're almost identical. They can talk to each other's receptors. Vasopressin's reputation is as a stress hormone and also a hormone of water balance. Now, stress, most people nowadays don't like this word. I certainly don't, but we're kind of stuck with the construct, okay? Fear, pain, these are primitive, and they're adaptive in acute conditions. Chronically, they almost never are adaptive. They create trouble. Vasopressin is a pro-inflammatory molecule. It actually has it can have some anti-inflammatory effects. Most of what it does is pro-inflammatory. It's a fast strategy of defense. Oxytocin, you can think of it as a fire extinguisher. So if you leave here with no other information, just look at this picture, which of course I borrowed from the internet and added my own words. Oxytocin mediates cues of safety. It helps to explain evolutionary biology, benefits of nurture, social support, co-regulation. Many of you, probably almost everyone here, has some interest in trauma. In 2024, actually over into 2025, the paper was published. We did a study. Alex Horn was a graduate student who worked with me. Alex is amazing. He's a major in the Air Force. He ran around the southwestern part of the United States, finding really stressed people. Some were patients with a diagnosis of PTSD. Others were people running marathons. He ran one himself to prove how hard it was, and he's super fit. He went to sleep at mile 82 or something. He said, I'm sleeping on my feet. This is crazy. But here's what their oxytocin and vasopressin looked like in our assay. They were about the same in this particular metric, or this particular assay. This is what the PTSD people looked like. They had almost double the level of vasopressin and half the level of oxytocin. Frankly, we've done lots of studies. I've never seen this kind of separation in anything else. So what does this mean? Individuals with a trauma history, in this case, they're diagnosed as having PTSD, probably have less access, at least this population, to the benefits of oxytocin and more exposure to vasopressin. And we could go through this some other day, probably a very hypersensitive vasopressin receptor. Now, I want to talk quickly about hormesis. We've each got just moments here to spill our stories. Hormesis was not a new idea. The philosopher Nietzsche summed it up saying, that which doesn't kill us makes us stronger. Now, my husband hates this stuff. He gets very upset because he wants me to tell you, and I want to tell you, it's all about whether it's manageable or not, okay? No pain, no gain is another way, though, that people have used to explain in the context, let's say, of exercise, what hormesis is. Now, this system is not truly balanced. It's always dynamic, always moving, but you have stress activation, and again, I have to use that word, sorry. You have increases in vasopressin. In a healthy person, they're going to be balanced by oxytocin, and the system will go forward. I want to show you, this is probably the most important part of what I'm going to say today. It's a little written in small letters, but the basic idea is hormesis is very popular in the wellness world. How many of you have heard of it before? Okay, so people who are deeply involved in things like exercise, cold plunges, intermittent fasting, temperature extremes, whatever, are very interested in it because they know that it's the stressor followed by a period of relaxation, safety, and recovery that creates the benefits. What's interesting, some of you are also interested in psychedelics. I spent about a year just combing through the psychedelic literature because, to be honest, it didn't make sense to me. Why were people making themselves sick to get better, okay? And the answer, that's hormesis. And what I started to realize, and there's not much literature, but basically almost every psychedelic has some initial phase of sickness, followed by a phase of wellness. They call this come up, come down. The same principles, by the way, apply to the GLP-1 agonists that are so popular now. There's a sickness phase. This is a clue, and it was Steve who actually told me about this years ago because he had some contact with NASA, and they were finding space sickness was caused by vasopressin, of all things, right? People were vomiting, and then hypothetically getting well. Well, vomiting, there's now quite a nice literature on this. Vomiting is vasopressin in action. You've got to fix it if you keep on vomiting. It's called hyperemesis. It's a very bad thing. But if you have a brief period of sickness followed by a restoration, you're probably seeing, and this is not well documented except fragments of knowledge from animals, but you're seeing a sequence of vasopressin, oxytocin. I think of it as kind of like a volleyball game. If you want to spike the ball, somebody has to set it up, and vasopressin seems to be the setup. Time and context matter. If you have an overreaction, you stay stressed too long, it isn't good, right? No shock. Okay, I've got one slide that hooks this all together, obviously in a 15-minute talk. We're not going to go through this in detail, but thanks to chat, we now have colors and beautiful things. On the left is oxytocin in so-called traumatized people. On the right, oxytocin's fixing all that stuff, okay? Now, I am not recommending you run out and get oxytocin and give it to your patients. Please don't. Please don't. Because this system is a real system, and it'll start to adapt. It won't necessarily do what you expect. And the research is, you know, uncertain at best, okay? Because they give oxytocin as an intranasal spray. That spray, A, may or may not get to the brain. B, nobody has bothered to take into account this hormetic setup that I'm talking about. But, and I won't go through this in detail except to say hydration is underestimated. I don't just mean water, I mean adequate minerals too, because vasopressin's very, very sensitive. So if you're on a high salt diet, you're eating fast foods that have been preserved, you may actually be treating yourself with extra vasopressin. This is all set up by early life experience. I probably don't need to tell you adversity in early life is generally a disadvantage later, but not always. So one of my colleagues, Bruce Ellis, keeps talking about adaptive calibration. If you live in a harsh environment, it may not necessarily be in your best interest to go down this take it easy pathway. There are many functions. We won't go through them. I want to say the whole body, every tissue in the body responds to both oxytocin and vasopressin, contrary to this bad reputation the hormone had as important to females only. It regulates the autonomic nervous system, which regulates everything. All right, what to do about all this, I am showing you. I love this. I don't own this slide exactly, but you get the idea. People are desperately searching for ways to balance their life, as is Yogi here. And all of this, frankly, it's often called biohacking. I think we are in desperate need of taking these systems seriously and figuring out how they work so we can use them. Oxytocin and the ventral vagal circuitry act as signals of safety. They help to explain what you've heard over and over at this conference, social safety. A sense of safety with others is so critical. What triggers oxytocin release? Well, you can look it up on the internet and find all kind of garbage, okay? Like 9 seconds of hugging. Okay, so if I run up to you, I'm a stranger, and I hug you for 9 seconds. Please, I don't need to say anymore. Okay. Sex is very good. Okay, because orgasm requires oxytocin. I won't go through all of this except to say babies are, if you could do nothing else and you really can't figure out how to adjust the system, loan yourself out as a baby holder in intensive care unit someplace. I almost guarantee you'll not regret it. Now, the last I have, I think I'm out of time, but I want to say one more thing. This is I don't have a conflict of interest, but this is definitely a plug for where the future is going. Steve and Anthony Gorey, who's back there someplace or was, in the back row, have designed a sound or sonic augmentation technology, a company called Sonosia that Anthony is running, has designed this. Donna Leah Gels here on the second row, Audrey, who's next to her. These folks, Audrey Dale, they have just published a paper in Frontiers in Psychiatry. I hope I got the right Frontiers, 2026. I hope I have that right. And this was done under considerable difficulty, just taking salivary samples before and after the music. It is, I told Donnelly, I said, this is against all odds. This is not going to work, but it did. And it's statistically at least significant before the music and after the music. It's not a huge increase. It was only, I'm sorry, I used the word music. Anthony, I'm sorry. I'm so sorry. After the sound, sonic augmentation technology. It's not really music. You can ask him why it's not. It sort of sounds like. But here's what happened. The people who were anxious got the best effect, the biggest effect. I think that's hormesis in working. Where are we going? If we start to understand this, we can change everything. I like pictures of babies, especially this one, because she's our granddaughter. And I like what Dostoevsky said, the soul is healed by being with children. If you don't know what to do, if you've got a personal problem, if you can help somebody else, especially a vulnerable person, it doesn't have to be a baby, but that's a way to help yourself. So I thank you, and I defer to the next speaker. Oh, thank you. Thank you. Here.
All right, can you all hear me? Okay, hello. Thank you for being here at the very end of the conference. I also first want to say that I've really enjoyed sharing this space with you all and learning together the past few days. My name is Hannah Swerbensky. I'm incoming research faculty at the University of Rochester, and today I'm going to be talking to you about biological embedding of stress and sharing insights from a landmark prospective longitudinal study of the effects of child sexual abuse on health outcomes and development. And we're actually lucky to have the PI of this study here today, Frank Putnam, truly a giant in the field. He's waving up here. And I'm excited to get started. So as a roadmap for today's talk, I'm going to tell you a little bit about just an introduction and overview to models of biological embedding of stress. I'll comment very briefly on why longitudinal perspective designs are a particularly useful research tool for testing and understanding these models. And then the bulk of my presentation is going to share selected findings from the Female Growth and Development Study, kind of a greatest hits, both of my work and other colleagues who have worked on this study over the years. And then hopefully we'll have time to do some science translation and talk just briefly about practical implications and pathways to healing. If you've been in the ACEs literature for a while, you may have seen this figure or a version of this pyramid figure before that essentially outlines a potential sequential process by which adverse childhood experiences ultimately increase disease burden and can even lead to early mortality. So it's well established that ACEs increase risk for chronic conditions such as diabetes, hypertension, obesity, COPD. But what I think is potentially underappreciated is that these associations between adverse childhood experiences and physical health problems hold even after you control for lifestyle factors such as drinking, smoking, nutrition, and so on. And I think this is really important to highlight in terms of destigmatizing people who are experiencing potential negative health effects as they age due to early adversity. And one thing that I think is potentially underappreciated, at least to the general public, is this earlier stage of the pyramid that relates to disrupted neurodevelopment and the process by which this early adversity becomes embedded in the body. When I was first starting my career, this is a figure that really stuck with me. I think it's one of those figures in science where a picture truly is worth 1,000 words. And what you're seeing in the top panel is a neuron from someone who grew up in a supportive developmental environment. And then in the bottom panel, you're seeing a neuron from the prefrontal cortex in someone who grew up in what McEwen here is calling toxic stress, other people might call chronic early adversity. And what you'll notice is that there's notably more dendritic branching in the top panel, so there's more spines coming out of the soma of the neuron so that more connections can be made. This picture is one example that illustrates what biological embedding of stress can look like. So biological embedding of stress refers to how early chronic stress becomes physically inscribed in the body, and this occurs across multiple biological systems, things like brain structure, gene expression and epigenetic modulation, or as I'll mostly focus on today, stress response system calibration. I want to walk us through this figure that can also help us understand why some people experience greater health effects than others due to early adversity exposure. If we start at the top of the figure, we see what represents the everyday milieu of life. So everything from your daily hassles, or what we're here to talk about today, potential experiences of trauma and abuse. And the way that we perceive those experiences of trauma and abuse are in part filtered through individual differences, so things like our genetic predisposition and personality, our developmental level, which also influences how we perceive stress and trauma while it's happening. So those differences from person to person, from client to client on your caseload in terms of their sensitivity to threat, feelings of helplessness, hypervigilance, which then influences behavioral responses, everything from fight or flight, freeze and defeat, as well as coping responses, which the clinicians in the room probably have more direct experience with than I do. But what I think I really want to zoom in on is the bottom of that figure, the physiological response piece, and I want to provide some definitions for what you're seeing at the bottom of the figure. So, allostasis refers to the process of regaining stability. When you experience something traumatic or stressful, your body wants to engage in allostasis to get back to baseline. But particularly when you grow up in a chronic adversity type setting, your physiological system has to adapt to that chronic level of stress exposure. This adaptation refers to long-term physiological changes due to sustained stress. But adaptation also comes at a cost. That cost is something we call allostatic load, which refers to the cumulative wear and tear of chronic stress on the body. And throughout this presentation, when I share research findings with you, the question I want you to hold in your mind is, what did this person's body learn, and in what environment did they learn it? So keep that question in mind. Next, I'm going to talk through the design of the Female Growth and Development Study. This is a 30-plus year longitudinal study with 187 female participants, half of whom were recruited after experiencing child sexual abuse. And I've listed the criteria for the child sexual abuse group here, and I'll note that they participated very soon after disclosing their abuse experiences, and specifically that we recruited for substantiated sexual abuse by a family perpetrator that included genital contact and/or perpetration. So in some ways it may represent more severe cases. Ethically, we cannot randomize children to experience abuse or not to study its effects. So the next best scientific tool is rigorous demographic matching. So we also recruited non-abused comparison participants who came from the same family compositions, racial and ethnic background, zip codes, to try to balance out those groups and get the closest we can to causal inference. We followed these women for 7 time points over 30 years that you can see laid out in this table with exceptional retention. And I also want to explain a little bit what I mean when I say longitudinal prospective design. So prospective means we were asking about their traumatic reactions as they were happening, rather than using a retrospective design where you ask someone to recall what their reactions were years before. And longitudinal means we have a degree of temporal inference. Lastly, this is a cross-sequential design. That means we recruited a relatively wide age range of 6 to 16 years of age at baseline that lets you do something statistically called arraying by age, so that you can see what happens continuously over development rather than little snapshots across the lifespan. All right, so this is an overview of the Female Growth and Development Study. And if you're like me, the first time I saw this figure, I said, damn, they really thought of everything. And that's part of why this study has been so prolific in its contributions to the field, resulting in over 50 publications to date. Obviously, we can't cover all 50 today. It's going to be some of the greatest hits. And before I get into the results themselves, I'll briefly mention that I think child sexual abuse is a useful traumatic event for studying biological outcomes because it represents chronic adversity oftentimes, and it does have the strongest association with obesity of all forms of child abuse and neglect based on previous meta-analyses. So a lot of the findings I'm going to present today, I believe, may generalize to other forms of chronic adversity, but that's something that has to be empirically validated. I also would be remiss not to take a moment to thank the principal investigators of this study, including Frank Putnam, Penelope Trickett, who's unfortunately passed away, and my postdoctoral mentor, Jenny Knoll. So the very first study I want to share with you, or first paper to come out of this study I want to share with you, comes from Trickett and colleagues. And she was interested in this question of how does child sexual abuse affect stress response system functioning over development. And she measured this using basal cortisol, which essentially reflects the levels of circulating cortisol in your body when you're at rest or just in your day-to-day life. And what she found when she tried to fit trend lines to the data, on the x-axis, you're seeing age from 6 to 30. On the y-axis, you're seeing basal cortisol levels. The red line, you're seeing the trend line for the abuse group, and the blue line is the non-abused comparisons. And so if you look at what's happening at age 6, we see that the abuse group has higher basal cortisol levels than the non-abused comparisons, but by the time they reach age 30, the trend lines cross over. And so what that is indicative of is something called HPA attenuation. So that means that in the immediate aftermath of abuse and disclosure, these girls were exhibiting higher levels of circulating cortisol, which in a way is the stress response system. It's a way of the stress response system to be hypervigilant or ready to respond. But by the time they reach their 30s, we see that crossover. They have a less steep slope that's indicative of hypocortisolism. And notably, we see these differences hold even after you adjust for their mental health trajectories over time. So we're coming back to this question of what did this person's body learn and in what environment did they learn it? This first paper would say that the body learned in response to their abuse experiences at first to be hypervigilant to potential threats in their environment, but over time that became unsustainable and they transitioned to a more blunted level of circulating cortisol or hypocortisolism. What I think is really interesting is that that pattern of HPA attenuation also helps explain some of the effects of abuse on health. So in this paper in psychoneuroendocrinology, we see that the association between child sexual abuse and obesity is mediated by that pattern of HPA attenuation. And what I think is remarkable is that this mediation pattern holds even after you control for minority status, parity status, steroid medication use, depression history, and disordered eating. And for the clinicians in the room, I'm not saying don't treat eating disorder symptoms, but I'm saying for the clients on your caseload who may be presenting with weight management issues or body image issues, it can be a powerful piece of psychoeducation to say it's not just what you're doing, some of this is happening on a biological level. I'm going to transition into another paper from this study that we recently published in Psychoneuroendocrinology, which answers the question of how does child sexual abuse-related biological embedding influence mental health trajectories. So in this study, I ran a growth mixture model, which is essentially a statistical way of saying, what are the different subgroups of trajectories of PTSD symptoms in my sample? And one of the groups we identified that I was particularly concerned or interested in was the chronic high PTSD group. So these are women who are experiencing clinically significant levels of PTSD from late adolescence all the way to midlife. That's a very major burden to hold. And what we see similarly here is that the effects of child sexual abuse on chronic PTSD are explained in part by that same pattern of HPA attenuation, such that HPA attenuation tripled the risk of experiencing chronically high PTSD, or if you want to phrase it in the most technically statistically accurate way, for each 1 standard deviation decrease in cortisol slope, which reflects greater HPA attenuation, we see nearly 3-fold higher odds of belonging to the high PTSD group relative to the low PTSD group. But I also want to say not everything is biology. So while the chronic high PTSD group's membership was best explained by biological embedding, I was also worried about this increasing PTSD group. And we saw the increasing PTSD group who had just kind of barely subclinical symptoms in adolescence that eventually became higher and as high as the chronic high PTSD group. Membership in that group was best explained by re-victimization, such that there was a 27% increase in odds of belonging to the increasing PTSD group per subsequent traumatic event reported. 27% might not sound that high, but many of the women in this study are reporting 10+ traumatic events over their lifespan. And then especially remarkable, we also saw that adult sexual victimization specifically was associated with a 5 times higher odds of belonging to the increasing PTSD group. So thus far, we've talked a little bit about biological embedding as a mechanism. I want to come back to this idea we mentioned earlier of individual differences, and specifically genetic predisposition. This is cheating a little bit because it comes from a different cohort, but I think it's relevant to the findings I'm talking about today because in a separate sample, we observed nearly the same different subgroups of PTSD trajectories over time, including that same prior group we identified, the chronic PTSD group, where they're exhibiting high PTSD over several years. And in this study, we were interested in how exposure to child sexual abuse and genetic predispositions may interact to shape mental health trajectories. In this case, we measured something called polygenic risk, which refers to the combined effect of many small genetic variants that collectively influence PTSD risk. So it's not a single gene, it's risk shared across many. And if I draw your attention to the top right panel, what you're seeing there is among the people who experience child sexual abuse, on the x-axis, you see their polygenic risk score, and on the y-axis, you see their odds of belonging to the chronically high PTSD group. And that's a linear association that we identified to be a super additive effect when we quantified the interaction. In contrast, the resilient group that exhibited low PTSD symptoms across the study period had a linear association in the opposite direction, such that as polygenic risk scores increase, the odds of belonging to the resilient PTSD group decreased. So if we zoom out conceptually, so far we've talked about how biology sometimes serves as a mechanism that explains how early adversity affects physical and mental health. And then in that last example, I showed you an example of how biology sometimes serves as a modulating factor or a risk factor for who will experience negative effects of adversity. I have one last study I'm going to share with you. This is another how question, so a mechanism question. And now we're back to the female growth and development study sample. And what I hypothesized is that there may be a multi-step process that leads to physical health burden at midlife, such that child sexual abuse exposure may lead to faster adiposity accumulation, so more rapid weight gain over adolescence and early adulthood, which in turn could increase chronic inflammation and ultimately lead to more physical health problems. And so what we see, this is our path model. I'm showing you just the significant associations, and I'm gonna walk you through it step by step. So this panel represents a lifespan from around age 11 on average at time 1 to around 37 on average at time 7. And the first part of what we see is consistent with previous research with this cohort. Child sexual abuse was associated with more rapid adiposity accumulation over time, so a steeper BMI slope. And in turn, a steeper BMI slope was associated with 2 out of the 3 inflammation markers that we looked at, so C-reactive protein and albumin to globulin ratios. We also see that BMI slope did exhibit some direct effects on more generalized health outcomes like blood pressure and overall subjectively rated health. But there were some pathways as hypothesized, particularly for respiratory problems, where increased risk for respiratory problems was carried through that pathway that we hypothesized from abuse exposure, more rapid weight gain, higher C-reactive protein, and then higher respiratory problems. I was overzealous about how much I could cover, so we're gonna skip this. But it will hopefully be out in the Journal of Family Violence soon. You could check it out then. I wanna wrap up with our key takeaways. So we've defined biological embedding. That's a process by which early adversity gets under the skin to affect biology, how early adversity is physically inscribed into the body. We've seen that mental and physical health consequences of adversity are not simply due to lifestyle choices, but a large portion of that burden is carried at the biological level. And lastly, we've also seen how risk and protective factors at the biological level can help explain who is resilient and who experiences negative health outcomes. So that also helps us allocate prevention resources and intervention resources. And if you take nothing else from this talk, I hope that in your work going forward, you continue to come back to this question of what did this person's body learn and in what environment did it learn it? I also know that a lot of my talk has focused on the negative health outcomes associated with adversity exposure. I do want to acknowledge that there are very talented researchers conducting work about how interventions can potentially reverse some of this biological embedding of stress. For example, Mary Dozier is doing work on attachment and biobehavioral catch-up and seeing a lot of promising effects for early intervention. And this is an area that I think the field is moving towards. And lastly, I want to conclude with what you as audience members can do to help alleviate some of this. So as healers, you can believe survivors, engage in evidence-based treatments, consider developmental history, and integrate with medical teams to try to coordinate care across systems. And you can also prevent future abuse from happening, such as by breaking intergenerational cycles, supporting primary prevention, and engaging in policy shown to reduce rates of child abuse. I think I can take one really quick question while Dr. Gonzalez gets her slides up. Thank you. I guess you could just ask, yeah.
Since there is a HPA attenuation, and we know that the pituitary gland is targeted into this, I was wondering because I found many ovarian insufficiencies, and I don't know if it's casualties, but.
So you're speaking more to reproductive system outcomes? Yes, so that is something we've also seen in other data with this study.
Repeat the question for people who are hard.
Oh, got it. So the question was about how this HPA attenuation may interface with reproductive health outcomes related to early adversity exposure.
Is connected with gonads.
Yes, true. So I haven't tested HPA attenuation as a mediator of effects of early adversity on reproductive health problems, but we do see in other papers from this cohort that child sexual abuse is associated with disrupted pubertal timing, such that the girls in this study experienced an earlier onset of puberty. So I do think it's a very plausible hypothesis and something I'd love to look at in future work, and I appreciate your question. Thank you.
Hello, hello. Hi, last but not least, am I right? Get it together, get a little amped up, a little more sympathetic than parasympathetic. I promise not to take up too much of your time. I really only have one study, and I'm actually gonna shorten my talk even a little bit more because I think heroes really need to be sung, and Sue Carter, you are not known as Mrs. Porges in my circles. You are known as Dr. Sue Carter, mother of oxytocin, as in the first of her name. I actually would not be here if it wasn't for Sue Carter. She is actually my academic grandmother, and you once said in a random talk that you probably don't remember, you were talking about how hard it was for people to take you seriously when you were interested in studying this, female hormone. And you said at the end of it, don't listen to people, do science. And I literally have that quote embroidered, and it is in my office right now. It's not even done yet, guys. I promise to embarrass you. So my academic mother, Jess Connolly, I told her, I was like, I need to embarrass Sue. So I asked her to say some words about Dr. Carter. And she said, so Jess Connolly is a molecular biologist and has done seminal work, or the seminal work really, on the oxytocin receptor gene. So Dr. Jess Connolly said, Sue is, without question, the most influential scientist in my life. She shaped not only the way I think about science, but also the kind of scientist, mentor, and person I aspire to be. Her curiosity, courage, and willingness to pursue ideas that others overlooked taught me that the most important discoveries often come from asking unconventional questions and having the conviction to follow the answers wherever they lead. What I admire most is that Sue never separated great science from generosity. She built a career that transformed our understanding of oxytocin, attachment, and social behavior, while also lifting up generations of trainees like me and colleagues along the way. Her impact extends far beyond her remarkable scientific contribution. It lives on through the countless people she inspired. That was off the cuff, Sue. So if you could just, I don't care about my talk. Can you guys stand up and just clap for Sue Carter? Thank you for everything that you've done. Thank you so much, Sue. Thank you. And guys, it is all downhill from here. So today I'm not going to be talking about any OT work, which feels like a crime. I should have somehow stuck it in. But I'm going to talk about developmental calibration of adult risk-taking, and in particular thinking about what are kind of the underlying neural strategies. Am I good now? Okay. The developmental calibration of adult risk-taking, and in particular thinking about this idea of neural strategies, that you can get to the same behavior, but you have sort of different underlying neural efforts, and that's going to be related to the match or mismatch between your developmental and your current context. I always like to start with a thank you. One, because apparently gratitude really lowers your stress response, so this is also very selfish, but also because it's true. Science truly is a team sport, and I couldn't do this without my wonderful lab who's here. The road ahead, I'm really just doing two things. I am a weirdo. I guess it comes with the lineage. I'm a weirdo. I think about context in very specific ways. I think about it as more of a behavioral ecologist than anything else. And then I'm going to go through one study, just one study, and do a deep dive to talk about, how does developmental context shape risk-taking behavior. Okay, so what do I talk about when I talk about context? This is a real picture of Rio de Janeiro, and you see these two different places where a child could, and children, could exist. If you view it from a stress model, you might start ranking and saying one place has higher stress than the other, and then you make predictions about what that means for their health. And that is a cumulative risk model, and it tends to be pretty freaking true from that standpoint. But from the standpoint of trying to figure out sort of like what is adaptive and how does biology try to keep you alive? Because oftentimes what biology is really doing, guys, it has a really low bar. Survive, procreate, that's it. Survive, procreate miserably, not ideal, but it'll take it, right? And so I'm constantly thinking about, I view this more as a set of problems. There's specific behavioral ecology problems, but I promised you I was going to be short. A set of problems that then people are flexibly figuring out how to solve. And not only are you seeing problems, but they're seeing different resources in this space. So it could be that on the left, you have, right, clearly high stress there in terms of literally temperature controlled, right? There's things that are very low level there, but you may also have a warm and supportive family. You may be getting that OT like crazy, right? You may have other resources that are not material. And on the other side, just because you're rich doesn't mean that you are not, at risk for trauma, right? You're not at risk for family abuse. And so these things can coexist. We'll have both challenges and resources in the exact same space. I'm going to skip a couple slides. I promise you don't need to know any of this. All right. Okay, so this is the way that my lab thinks to think about how to process context. For one, we really think about environmental pressures, both from like that physical affordances sort of space, so literally physical space, but also what are the blockers there, and what are the dangers there, what is the predation, for example, and then also the social aspects. What are the social affordances? Who is around you? Do they meet some sort of need? And on the other hand, are they the source of some kind of, in this space, I can just say trauma. From that, the developing body learns sort of how to solve this problem of the things that are around them, in which case here we're tuning, right? We're calibrating, as the great Bruce Ellis would say. We're calibrating these systems. In this case, how risk-taking should I be? In a context in which I'm not sure when the next reward is going to come, maybe I actually need to risk some things in order for me to get it while the getting is good. But that might not be a good strategy in another context that is much more predictable. This is aligned with then changes in the actual central nervous system, right? In order to support these kinds of strategies. And not only are there behavioral strategies, but even neurally, you'll have different sort of neural strategies that arrive at the exact same behavior. For example, I may have a pretty low-level risk-taking behavior, but for me, I'm really working hard in order to inhibit myself, whereas for someone else, it's nothing. It is the thing that they are used to doing. So this asks questions of what are sort of the resources and challenges in an environment? How does that then lead to this different sort of behavioral differences and then these central nervous system differences? In order to investigate this, those of you who know me, I'm really big proponent of we need to get out of the college space and need to be in community and get neuroscience data from representative samples. But in this case, the actual ideal people to look at this, right? If I want to look at what is a population that ended up in the same space, but actually came from very different, right, spaces, I look at a university. You have people who are coming from all these different walks of life, and yet they are ending up at, in my case, or in this case of this study, at Cornell University. And so one of the things that we want to characterize is who are the students who are coming to Cornell University? What is their developmental context, their resources? And spoiler alert, one of the things that I found from a big, huge study that I did is constantly I would see either they're really rich and socially disconnected, or they are relatively poorer, but they have really, really rich social connections. And what does that do? What does that research, what does that asymmetry sort of do? Okay. So kind of question is, do people count on a known resource for risky decision making? If I'm used to a social resource, is that what I count on to sort of decide at what point should I be risky and at what point should I actually be a little bit more risk averse? So the premise is individuals capitalize on resources, right, to solve the kinds of problems around them. There's a hypothesis called sensitized specialization that basically says the traits in adulthood that traits show up in adulthood in the same context that they were learned previously, right? So you can do different things at different times. You're a very flexible human being, for goodness sake. But if I put you in a context that is similar to your childhood context, you will act more like that, more like you were before. And I hit this. So the strategies may lead to the same behavioral outcome, but incur different sort of biological costs depending on where did I learn, for example, how to have good inhibitory control during risk-taking behavior. So again, we looked at this college population, which had different kinds of developmental background. These are 43 cisgendered folks, mostly around 20, generally ranging in different parental incomes and I'll say somewhat ethnically diverse. We looked at their developmental context, both in terms of economic and social privilege. And then we also looked at the current context in terms of like how much social contact they have, any kind of issue, motivational sort of phenotypes related to behavioral activation as well as depression to answer this question. We also looked at a task called the balloon analog risk task, which is often used to essentially try to model how willing you are to take a risk in order to gain more resources, but possibly risk losing it all. So in this case, whenever you see the red balloon, every time you pump, the balloon grows bigger, and then you get more money. Yay, reward. But you don't know when. At any moment, that balloon can pop, and then you lose everything for that round. You then have another condition with the white balloon where you pump. This is a condition I added. And you keep on pumping, and eventually it'll stop, but it's a guaranteed reward. So the red balloon is a risky reward, and the white balloon is this kind of guaranteed reward. And then we have this neutral condition where you're pumping, so it's all motor, but there's no reward in it. And from that, we are able to do various things. One, at the behavioral level, we are able to use a computational model to get at this trait called loss aversion, which essentially means like, how badly are you stung after you lose? So if I popped that balloon, lost all my money, next time, do I then only pump 3 times? Because maybe, you know, twice shy or whatever the term is. So I'm going to change my behavior. And that is different. Some people are highly sensitive to experiences of loss, and others are like, nope, I'm gonna keep being risky, right? That's actually me, sorry. Okay, so you're able to see behaviorally this idea of this trait of loss aversion, but then through essentially different contrast, you're able to see in the brain what's going on when they're in the risky condition versus the neutral condition, or the risky condition versus the guaranteed reward, which is very nice because it takes away the reward sensitivity aspect, and it's really just like, how willing am I to take a risk at this very moment? So the aims of the study are just 2. One, we want to cluster participants in a bottom-up way on developmental affordances. But again, I had seen like a giant swath of people in a separate study, and I was like, I'm pretty sure it's gonna end up this way, because that seems to be the Cornell student. And then 2, I'm trying to see whether we can dissociate sort of the behavioral aspects, again, assuming, hey, you ended up here somehow, right? People who are coming from very different places, you ended up here. Therefore, I have this belief that you all must, to some extent, share this trait of risk-taking and other factors. And yet, there might be different sort of underlying neural tactics for this. Okay, so I will orient you to every single graph, I promise. So here. So on the bottom is essentially the social and economic resources, and then you have the Z statistics. And what you'll notice is, as I said, we have individuals who are economically rich but socially not as good, and those who are socially rich but economically not doing quite as well. And in each one of these, you'll see a difference. What's interesting is, right, these are the measures where these clusters were distinguished in a multivariate way, but we didn't see any differences in terms of parent-child relationships. The ACE scores were also used, no real big differences there, or depression or behavioral activation or inhibition. So really these folks are differentiating themselves on this sort of socially or economically rich dimension. In terms of how they're doing in reaction time for that balloon task, there's a main effective condition. During the risky thing, your reaction time is just slower for this. And then in general, you're getting more money in the guaranteed reward condition than in the risky condition. I think we expect that. No differences between groups. When we look at the whole brain and we look at risky versus guaranteed reward, we see a canonical sort of reward-related pattern, lots of midbrain, ventral striatum, and then also the ventral tegmental area. Wonderful. Okay, this is just to figure out if the task worked. Then I said, right, we looked at them in terms of loss aversion. No difference between groups in terms of loss aversion. However, when we look at the brain, we also see that as loss aversion increases, you also see this increased activation in the supramarginal gyrus. And the SMG is this place that is constantly that seems to be pretty imperative to doing that inhibitory response, so like a go, no-go sort of trial. And it's also in contact or in connection with other features of the brain that allow for that inhibitory sort of response. So the more loss averse I am, I'm sort of hurt by, or I change my behavior a lot based on the experience of a loss, the more likely I am to like really charge this. I'm going to use this a lot. Okay, but we're interested in the groups. What happens with the groups? So the socially rich, that's blue, and then we have the red and the ER. So let's look at the blue on here. On the bottom is the loss aversion, and then on the y-axis, you'll see essentially the neural response to that risky condition. Boom. All right, so what do we see? So for the socially rich group, as loss aversion gets more intense, you also see this increased sort of neural effort that is necessary in order to arrive, again, at the exact same sort of response. We also looked by a pump by pump. So in this case, we're seeing this sort of dissociation between behavior and neural effort. All right. What about if I then change, I look into the current context, right? How about for those people who are socially rich in their development, what if they have really great, awesome social support while they're in college? It tends to happen less because from another study, I do know the lower your sort of neighborhood quality, the less sort of belonging that you feel when you're in college, especially a place like Cornell. But let's say you did. You figured her out, you're at Wadi, you're at these other wonderful groups that are meant to make you feel like you belong, and you have tons of social support. Well, again, social resources are at the bottom, and then we see the % signal change in these regions of the brain. And what do we see? So looking at the blue line, as the social resources increase for that socially rich group, they don't have to work as hard in order to arrive at this, sort of less risk-taking kind of behavior. And I think that's pretty neat. Well, what about on the other side? What if I looked into economic kind of resources, and then I could look at the economic group? It didn't pan out. Okay. But we can think about other things that might be related to that economic group. So let's hold a thought. So some interim. Don't worry about that seed. So what we do see is that higher loss aversion, which is related to risk-taking, coincides with this greater bold signal in this particular region. This socially rich group has this higher neural response to the risky condition with higher loss aversion. However, if you increase the social support, then you don't see that same level of effort. That effort starts to go down. So this is a one-sided mismatch story saying that as soon as the socially rich group has that social, that sort of developmentally matched resource, then you see that reduction in effort. Okay. So we know that like the SMG is not the thing that's doing everything. There's high connections that are necessary in order to actually create behavior. This is complex behavior that is happening. So one of the things we can do is we can look at connectivity with this SMG seed to try and understand better other factors that might be changing, other neural tactics that might be going on as a function of, you know, what your developmental context is and then what your current context is. So in terms of connectivity, this is kind of very canonical for, again, inhibitory sort of or risk-taking kinds of studies. You see that the SMG is highly connected to these other features of the brain when in the risky condition versus not in the other one. Okay, cool, it's working. But what about when we add the developmental context and the current context? Bear with me, there's going to be new colors available. Okay. So essentially, we have to do a two group bicovariate analysis. On the green, you're going to essentially be seeing the neural areas where where the social resource group has a greater response to this incongruent resource. In other words, there's low social but higher economic sort of resources around them. And then the opposite for the brown. So go with me here. So think about it as incongruent. Okay. So here you see in the blue, as sort of the incongruent resource goes up, you're seeing this kind of increased activation, which is weird because it's like, don't you want more economic resources? I mean, I know I do, but still you see this weird thing where you have this higher neural effort. And then you switch it and you look where the ER group is actually more active. And you see on the bottom is social resources, and as social resources increases, that economic group now is working harder. So again, this weird mismatch story that I'm deeply uncomfortable with, because don't you want more social resources? I know I do. But it's interesting because it really all arrives in this particular dorsal medial prefrontal cortex area. So again, these are the regions where you see sort of like the effect of mismatch, but both are apparent in this DMPFC area, suggesting that there's some sort of compensatory response when you're in the resource incongruent condition. There are, however, especially for this, huge limitations, right? We really only looked at people who had successful strategies to get to where they are. In this case, there are many other strategies that could be represented on here. These are retrospective measures of childhood, so perception could be doing more of the work than the actual material consequences of that time. The sample is very small, and also we looked at a monetary task, which may be particularly meaningful. If we did a social task, might we have found something different, especially with that economically rich group. But for you, we really want the so what, but big caveats, okay? One, I think if I can sort of throw anything out there is that measuring behavior is important, but behavior alone doesn't tell you the cost of that behavior, right? We may all be able to, say, self-regulate in a meeting when somebody's saying something super sexist, but some of us are working harder than others at having the exact same behavior. And it's important for us to figure that out. Second, again, you arrive for this particular group, the socially rich, but economically poor students are arriving at the same behavior with greater cost. That sensitized specialization sort of hypothesis predicts this, that there's going to be ease when a behavior is done in the expected context, and there's going to be friction when it's done in an unexpected sort of context. So possibly, matching an individual to a resource that they're used to in order to facilitate some sort of change of behavior might be one way in which we can adapt this. So if somebody is used to, for example, and very good at for some reason, right, so using social support in order to change a behavior or cope with stress, then we might want to, one, assess how those social resources are going, and two, facilitate ways in which we can increase those resources. And the idea would be that that might be particularly well matched for that individual in a way that it might not be for another one. I'm done. So did I do it right? Good. Do we have time for questions?
We have 2 minutes.
We have 2 minutes, time for one question. It can be like, how's your coat so awesome? No, just kidding. We could also just go home. Okay, we don't. Oh, I got one question.
What were you most surprised about?
Wait for a second, 1 second.
Oh, sorry.
My question is for you, like what were you most surprised of finding?
I think the surprising thing is, I think it's pretty, for most of us, we're like, more resources, more good. And we just assume that resources are equally as good for everyone, no matter what sort of the background is. So it is a little bit odd to me to see that people are working harder when incongruent resources are higher in sort of their particular context. I think that is the oddest sort of outcome that I saw. And I don't really know what to do with it, honestly, other than to feel weird about it, pretty much. We good? All right. You guys did it! Woo! Thank you.